And again, go to module. And here I give you a pre-lab guidance. Again, here it's going to be where you guys are going to be clicking. And here's the pre-lab guidance. I'm not going to talk about it. You guys can read it on your own. But this is what you should have: data, your date, your course number, and the title. Objective pretty much going to be the same for each experiment. I'm going to ask you to write it in 50 words or less. What's being investigated, what chemicals are being used, the techniques that's going to be used. Then this is the background information in terms of that. You guys are monitoring pH. What exactly is a pH? Okay. What does P and H stand for? So you got to bring the readers up to speed. You know that you guys are going to be studying acid-base chemistry. So what are acids? What are base? What is the chemistry that's involved? You really got to go back to some of the fundamentals and assume that your reader really is not well-versed in chemistry. So you got to bring them up to speed. So this is the background information. We're going to be looking at the pH probe. You guys are going to be, or the video is going to show you how it's calibrated. Why do we need to calibrate the pH probe? Okay, why can't we just use it out of the bottle? Okay, what is the importance of that? And then the data table that's going to be generated. You guys have test solutions. You got chemical substance. Test solutions like 1, 2, 3, 4, 5. Chemical substance like vinegar, ammonia, 7-Up, things like that. Then you're testing it with red litmus and blue litmus. If you watch the video, I actually show you those results in the video. You're testing it with hydroion paper. What is the color of the hydroion paper, and what is the reading based on that color? You're putting universal indicator. Okay, when you put drops of universal indicator, it will change color. I give you a legend so you guys can estimate what the pH is, and then you have your pH using Vernier. Trial 1, trial 2, trial 3, and the average. That's the data that's being collected. You will see that in the video. You have safety. Again, you just do like this right here is SDS for vinegar. Again, just do keyword SDS vinegar, SDS ammonia, SDS lemon juice. Some of these things you might not find in SDS because they're common household chemical. But try and do a little bit deeper search. Like for example, you guys are going to be using, I think, some of the other chemicals in here. I don't think cabbage juice are that dangerous, but try and put down some information. The quantity that's being used again, and then the disposal. All these are chemically safe, so you can just put in one container in terms of the disposal information. And that's basically what you would do in your pre-lab. Again, in this particular case, what you would do is you're going to write everything, your pre-lab, your data and observations, and your post-lab in this one page. Click on submit, right? Okay, pre-lab, you can start typing in here. And, or you can paste your Word document. If you decide to write in a Word document or Google Docs, you can just paste it in here. Okay, if you want to paste a screenshot, then you go over here and you upload an image. Okay, so you can upload it here. If you want to upload a screenshot of a data table, then this is where you would do it as well. If you want to write an equation so that you have Greek fonts, then you can do it here under the different types of formatting. Okay, you can have different formats. If you want to generate a table, you can generate a table here. This is where you would write your mathematical equation. So the text editor in Canvas gives you the flexibility as if you were writing a report on your notebook and you want to use that. Okay, let's face it, we're all going to go into the digital economy. Even though this may not be the program that you use in your career, it makes you get used to different ways of using whatever programs available and being flexible enough to use the tools that are provided. That's the whole idea. Okay, that's the whole idea here. So and then you want to submit, And then if you need to resubmit, then resubmit. And remember, when you resubmit, it'll give you the original document and you can modify and change it up to the due date. So I'm going to cancel this. I'm going to get out of student view. Okay. And then I'm going to show you exactly what I mean in terms of what you should be doing with your pH. So let me get a data set here. I'll show you what you would be doing. So these are my different substances, okay? And these are the average. This would pH average. So I'm at a point where I have the average pH. These are it. I have my universal indicator, what the pH are. I have my hydroion paper. You're not going to get a pH on litmus paper. Litmus gives you whether when it's red, it's acidic. When it's blue, it's basic. If you have red and stays red, if you have blue and it stays blue, it's sort of neutral. Okay, so you're not going to get a number from litmus paper, but you will get numbers from your pH readings, your universal indicator, and your hydroion paper. What you want to do is you want to take an average. So again, this is partial data set of how and you guys are going to generate a chart like this. Okay, so here you would put like maybe lemon juice. Here you would put like lye. I'm just making these things up. Here you would put like 7-Up, depending on the item. So write a description in here, and then I'll just leave this substance 4, 5, and 6. What I want to do next is I want to take an average. And you do that by typing in equals average, left parentheses, and then you want to highlight the cells in which you want to take an average of. Click on that. So the average of all these data points is about 2.18, okay? In order to change the number of digits after the decimal point, you can go over here, okay, and change that. I want at least 2 digits after the decimal. So I've changed the format here so that they're consistent. And I also want 2 digits after the decimal in the average. Again, remember the propagation of calculations. Click on this cell, go to this right corner, drag it down. Now I have the average for all 3. Now I'm going to do a column graph, highlight these, and then go to insert. And then you want to do a column. I just want to do this. I want to do 2D column. I don't want any of these fancy columns, just 2D is fine. And when I do that, look at that. It sort of groups it based on the names. Lemon juice has this. These are the first pH. This would be the universal indicator. This would be the hydroion paper. And this last yellow bar is the average. Okay, so what I want to do, however, is I want this to be increasing or decreasing pH. So what I'm going to do is I'm going to do this, highlight everything, and then I'm going to go to sort. So if you go to sort or data and sort, I'm going to go in here and I'm going to sort it by average. And I'm going to go from largest to smallest, okay, under sort. And then I'm going to say that it has a heading. It has a heading. And I'm just going to say, okay. See how that instantly changes as I do it on the fly? So that, again, that's the flexibility of Excel. If I want to do this and make it smallest to largest, I can do the same thing by going under data, sort. And now I want to change this from smallest to largest, and I can do that. And basically, this is the chart that you guys are going to be generating for your report, okay, in the processing. You're going to have the different pH, and you guys are going to show in a graph the different values, pH using Vernier, hydroion paper, universal indicator. It's to get you guys practicing using the pH probe, because we do use the pH probe a lot in this class. Okay, so that's basically what you want to do. Here's I think a copy of some of the items that we have. This is just a two data set. you actually want to do all 3 data set: Vernier, hydroion paper, universal indicator like this. Okay. So when you watch the video, you guys are going to be generating a table like this, and then you're going to be generating a graph like this, and then you're going to put it in, well, the data table is going to be in your data and observations. The table will not. The table will be there when you process the data. When you process the data. And then of course you guys are going to be talking about the data. You guys are going to be talking about different pH for the different substance. If you are surprised about the pH reading for certain things, or it's as expected, that's really what I have for you guys today. Again, if you go back to Canvas, so here we are in modules. Go to this part right here. It's the only thing that you should be working on. And I already given you the guidance. Okay. This part right here, the pH and the concentration of cobalt chloride, you guys can get started on that on Wednesday. Spend most of your time the next couple of days trying to do this part based on the guidance that I give you. This part right here, you can do Wednesday and you can turn it in up to next Monday, I believe. Okay. I think if I click here, it'll say March 1st. Let me get out of this view real quick. Leave student view. Yeah, it's March 1st until date. Okay. The due date is the 26th of February, which would be this Friday. If you want to get bonus points, the final date is next Monday for this report. Now, let me just mention before we all leave that you write in one space the report for intermolecular forces of alcohol. You write in one space the pH of acid base, but you write in 3 spaces in its different parts, just like you would do a different experiment. The 3 sections of cobalt chloride, the PreLab would be here. You write it, okay, by clicking on submit, and that will give you access to the text table. And you want to just put your PreLab here, and then you want to write in one space, in a different space, the data and observations. That's this right here. And then you want to write in a different space the post-lab. And that's the way the write-ups will be in your regular experiment. You have 3 spaces. I'll give you 3 links to put the different sections. And they'll have different due dates. They'll have different due dates. Just like you see right here, there's the PreLab, there's the data and observations, and there's the post-lab. The first 2, you put it in one space. But for this one, I want you to get used to writing the report as would be required when we do our first experiment. So that basically is it. I will go ahead and record, download the video for Zoom and then upload it here, upload it in Canvas later. But are we doing the worksheet too? The worksheet will give you a guidance. Don't turn in the worksheet. What you guys are doing is you're using the worksheet to write up the report based on the guidance that I provide. So no, don't use the worksheet as your template in what information to write in your report. So today we are just going to wrap up activity 4. I'm just going to show you what I've put in to guide you with part B, pH of test solutions. These things are called test solutions, the household substances. So in the procedure, you guys are going to see it referred to as test solutions or household substances. We'll talk about that. I'll show you where to find the guidance for that. And then the second part, or the last part of the 3-part series, remember these are 10 points apiece. So, And you don't have to fill out the worksheet. What you want to do is use the worksheet as a guide to how to craft your write-up. What we want to do is make sure that you practice writing up the 3 sections in your lab report. That way you have practice. That way, when the real McCoy comes where it counts 50 points, that is, and then 100 points for your evaluative report, then you can get the most benefit from these exercises. This is the second part of the 3-part series. And again, if you click on this, it will tell you right at the bottom, it's due on the 26th. You don't have to turn it in until the 1st. I think that's true for both. We'll visit this page again. Let me go back to module and take a look at the second part. That's the SpectroVis portion. Notice that in the third part, I break it up into the sections. The PreLab, and in the PreLab, again, regularly due on Friday, but you don't have to turn it in until the 1st. If you go to the data and observation, that's a different section because I want you to write it in 3 different sections, because that's the way you guys will be writing up your experiment for the regular reports. You're going to put it in 3 sections. Here, again, 26, and then you don't have to turn it in until Friday. And then we'll go to the third part. That's your post-lab write-up. Remember, these are just activities. They're not your formal experiment. So these things have a quicker turnaround. Your formal experiment, you have a week and a half. This is, again, Friday. You don't have to turn it in until the 1st. Okay. So you can always take a look at the deadline on these things, especially if you go to your calendar. I believe if you go to your calendar, it sort of summarizes it. Of course, I have a lot of classes. So what I need to do is I go to the right here and make sure that I'm not in the lecture, I'm in the lab. If you click on that, then it tells you exactly what's going on. And I think if you click on any one of these, it gives you the details of each one of these, okay? So use your calendar as a way of figuring out what's coming up. And then I think that if you go to, let me get out of here real quick. Usually if you go to home, you see a bunch of things that are coming up right here on this part of the menu. And again, that will give you an idea of what assignments are coming up. And I usually put something on the announcement page. Okay. So I think I've taken care of some of the questions that you guys have in the chat. Let me go ahead and talk to you guys about part B and part C of this experiment. And that's all I really want to talk to you guys about. Next time, I'll talk to you guys about experiment number 1, the kinetics of crystal violet. And it's important that you pay attention to experiment or activity part C, the SpectroVis of cobalt chloride, because the SpectroVis is the Vernier hardware that you guys will be using in experiment 1. You guys are going to be monitoring the absorbance of crystal violet. It starts out as a purple color, and then it sort of decays to a clear color, and you know the speed of the reaction based on the disappearance of that purple color. Okay, so that is experiment number 1. But let me go ahead and talk to you guys about part B and show you what we are looking for in that particular activity. So go to module here, and then go to, get rid of these right here. Okay, go to module, and then go to, let's see, get rid of this real quick. So we've already talked about intermolecular forces of alcohol. Here is part B, pH of household chemicals. This is a Zoom session from last time, what I will do is I will take our Zoom session now and append it to this. So it's just one clip. And I'll tell you in the announcement at what time to look for the second part, the part that pertains to the discussion we are having now. And then I'll put another clip for, I'll break up our Zoom session. So there's another clip for the cobalt chloride procedure. That way it's not all in one long clip. Anyway, let's take a look at this. And here are the guidance that I provide for this experiment. Again, I'm giving you, you have your training wheels on, okay? So that you know exactly what I am looking for in these reports. And hopefully I will wean you off and you will be on your own in terms of what to write down, but I need to give you some guidance. Here's the PreLab. You can click on that and I tell you, okay, always start with a heading. The objective of this experiment is very similar to what we had in part A of this activity. It's going to be generic, right? About 50 words or less. What the objective of this experiment is based on the reading of this procedure. What is to be investigated, chemical reactions, if there are any, or the chemicals. In this particular case, we're going to be studying certain chemicals, and we're going to be studying the pH levels of these chemicals, and then what type of experimental technique are we going to be using. And we're basically just going to be measuring the pH based on the Vernier probes and test papers. So that's what you're going to include in this particular objective, something that's to the point. And then the background information, I tell you, define what pH is. We take for granted what pH, we think we know what pH is. You guys should have enough knowledge from general chemistry and Chem 152 that pH measures the hydrogen activity of chemicals. So you guys are going to put that in. You guys are going to define what is the chemical phenomena that causes something to be acidic or basic. And you can use the definition, like the Arrhenius definition or the Lowry-Bronsted definition, that an acid is a chemical that releases its hydrogen. The more readily it releases its hydrogen, the more we give it a character of being a strong acid. The less readily it's going to release its hydrogen, we give it a character of being a weak acid. But as long as it releases its hydrogen, we consider it an acid. A base, on the other hand, is a chemical that is going to be a proton acceptor. It's going to take in hydrogen. The more willing it is to take in the hydrogen, the stronger the characteristic called basicity. Okay, the weaker it is to attract that hydrogen, then we consider it a weak base. So these are the things that hopefully you guys covered in Chem 200, but we are going to cover it in more details, I believe, in chapter 17, when we start to talk about acid-base chemicals. Because what you guys have been exposed to in your previous chemistry is just strong acids and strong base. The chemical will give up hydrogen or take in hydrogen, and it's an on-off switch. What you guys will learn in this course is that it's going to give up hydrogen, but it can do so with certain degree. It can give up hydrogen almost instantaneously, or depending on the environment, it'll give it up depending on to some degree, 10%, 20%, 30%. These things are called weak acids, or it might take in hydrogen to some degree. These are considered weak bases. So you guys are going to be talking about that. You guys are going to be talking about the probes that you are using to gauge acidity and basicity of these test chemicals. Okay, so you need to write a little bit about that. You guys are going to be using test paper, hydro-ion paper, litmus paper, and you guys are using indicators, chemical indicators that changes color depending on the acidity or basicity level or the pH level. The ones that you guys are going to be using is universal indicator. Okay, so that's what's in here. And then you talk in the procedure, you're going to be talking about the steps that that takes place in terms of monitoring the pH. You monitor the pH based on the pH probe and test papers on these test solutions. You also need to discuss why the pH probe is calibrated. Okay, we calibrated at 4, and calibrated at 10. That way we have an N range, and then we test at 7 right in the middle. So that's what this is. That's what this part is. I tell you, I give you some guidance in this description right here. And then, of course, the safety. What are the chemicals that are being used? And yeah, something like 7-Up, for example, there's multiples, a number of acid in that. If you ever take a look at the active ingredients of these soda beverage, it listed in terms of the most abundant first. So just Google 7-Up if that's what we are testing, and look at the ingredients, and the ingredients should contain like citric acid, phosphoric acid, okay? Depending on how high up in the ingredients list, that would be the active acid you guys are testing for. Chemical like vinegar, the active acid in that is acetic acid. Chemical like lemon juice, the active acid in that is citric acid. So these are the things that you want to write down. Again, more detail than just saying lemon juice, detergent, ammonia, active ingredient ammonia is NH3 ammonia. And then if you can, if they have an SDS page on that, then go ahead and put it on here. If not, don't worry about it. I will be able to double check whether or not there are an SDS page on these things. And then, of course, any pre-lab questions that are part of this procedure. I think in all these activities, there are no pre-lab questions, so you can just skip that. But when we do get to the experiments, there will be pre-lab questions. So again, I'm priming you so you know the format and the order by which you are going to write your report. Okay, that's the whole idea here. It's a little bit more work than used to. You're not just filling out a data sheet, but the data sheet really doesn't give you practice in writing up report. The data sheet and you guys are not doing the experiment. It's not like you're in lab and doing these things. You're just watching the video, and you can always scrub, fast forward, or go back if you miss anything. So what we're trying to do now is I'm trying to align the work that you do early on so that it helps you down the line. It actually will make your report better. And if your reports are better, you get a higher points on your report, which will translate to higher grades. Okay. That's always what I want you guys to get out of these labs. I want you guys to hone your skills in communicating what you have done in the lab. And if it means that I give you guidance early on, it's a little bit more work in terms of understanding the experiments, writing up the experiments, then invest the time so you can be better when it does count. These are just 10 points, okay? These are just 10 points. And I'll probably be lenient on these reports, but I do want to make sure that I give you back the feedback so that you don't make the same mistake when we get into the regular experiments. So that's basically the data observation. And then if you click here on the processing and then the discussion, I tell you exactly. And again, I use the worksheet as a guide of what to include in processing the data. Here I tell you that, yeah, you guys have a bunch of data that you've collected. You've basically completed what the colors of the different tests papers are, as well as the color of the universal indicator, and then the pH value based on the pH probe. And then you're going to kind of come up with an average and then fill out this sheet. It's in your lab report. So you have an idea of what the hydronium concentrations are and the pH value. Remember that you calculate the hydronium concentration from the pH by taking 10 to the minus pH. So if you have a pH of like 4.5, if you raise it to the 10 power, 10 to the negative 4.5, you get your hydroion paper, you get your hydronium concentration. Look that up in your Chem 200. If you're still unclear, I think I give you some examples in the lab manual. And then you're going to create a table, just like we did last time in our last discussion. And then you're going to make a column chart. And in this particular case, this is a simple column chart, and this shows just one pH value. But because we have 1, 2, 3, 4 columns here, or 4 set of data points, you guys will see 4 columns for one test solution, just like we did in our Zoom session last time. Okay, but you guys are going to be generating a column chart. It's not just the substance and the average, it's the substance, the pH versus of the probe, the universal indicator, the hydroion paper, and then the average. Take a look at last time's Zoom session. Then you're going to always create a table of results where you summarize what you found. Here, I tell you what should be included for this report, and then talking points that makes meaning of this experiment, and then taking this result, okay, and then discussing what that result mean. And then if you look at the lab manual, these were the talking points for that experiment. So you want to embed this in your discussion. And then write a conclusion. A conclusion basically is that you were determining the pH of some test solutions, and you determine the pH by a number of means, like a pH probe, the hydroion paper, the universal indicator, and you were able to test whether it's acidic or basic using litmus paper. There's 2 types of litmus paper, red litmus paper and blue litmus paper. Litmus paper is basically a dye. They put that dye on the paper, and depending on how that dye reacts to chemicals that are acidic or basic, the dye will, if it's red, it'll turn blue under basic conditions. It'll stay red if it's acidic or if it's neutral. The other dye of litmus paper is blue. Blue litmus paper will turn red under acidic conditions, but stay blue under basic conditions and stay blue under neutral. So the only way you determine neutrality is when red litmus paper stays red and blue litmus paper stays blue. Okay, so those were the types of tests you did for this particular lab. And then, yeah, that's basically the conclusion. And then you were able to rank the acidity, basicity level of these test solutions. So that is pH of acid-base household chemicals.